السلام عليكم ورحمة الله تعالى وبركاته. اسمي أنا قاسم محمد. معكم جهاد, Haya and Shazia. We will all give you the Pacademia. I will do sections 1.1, 1.2, and Jihad will do 1.3. Haya will do all of chapter 2, cells. And then I will come back and do 3.1 and 3.2, and then Shazia will do, you said 3.4, yeah? We will all do 3.4. We might eventually later do 3.5 for you as a recording, because you know, like, it's on the quiz and we are quite short on time. Okay? Did you all study a little bit? Did you like read the chapter? Yes. Did you like focus with the doctor? Again, that's fine. That's completely fine. We are here to like sum it down and you know like point out important stuff, difficult stuff, and clear up some like confusing things. Yo, Bismillah. Please.
The recording is starting. Please, everyone silent.
Just لو سمحت انا قاعد عم بصيح كثير يعني ما بدي صوتي بس لاخلص. فشوي على الحكي والامور. لو سمحت. يلا بسم الله. 1.1 Properties of life. Now, I'm not going to spend a lot of time on this. I'm just going to be really quick. Life is complex and dynamic. What does that mean? Life is all organisms are made from biomolecules. They're all carbon-based. All of the biomolecules are made from 6 atoms. What are they? Carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus. Sahel. Life is organized and self-sustaining. Life, it has like a hierarchy. You know, you have cell, tissue, organ system. So like the main thing to just point out, monomers will form polymers. So amino acids form proteins, monosaccharides form polysaccharides, and then nucleotides form DNA and RNA, nucleic acid. 3, it's easy. Life is cellular. Cells make up all of the living organisms. Life is information based. DNA is the code for all of the proteins that you have. Simple. Life adapts and evolves due to external stressors, external stuff. You will get a mutation which would eventually lead to some form of like evolution later on. This is all really simple. خلص. 1.2 Biomolecules. What is the functional group in this compound?
Raise your hands. We have candies. We have
guys
stuff.
لو سمحت. look. يلا يلا فاقت الناس الله اكبر. يلا مين حد؟ ها تعزم. بي وينه؟ Can you put them up?
OH is the hydroxyl. COOH is the carboxylic.
صح. اعطونا. تكمل؟ صحيح. Hydroxyl and a carboxylic acid. Now this is a question out of your book. It's the last question. Okay? So I highly like check your book questions. They help a lot. Now these are all of the different groups that you have to know. I'm not gonna go through all of them because you know time and stuff. So صعب صراحة. But I will point out a few things. Aldehydes and ketones are isomers, so they form longer. So they can all form longer structures later. So what makes a functional group polar? Hands.
Chips. Yalla.
Huh? Can you expand on it more? Why oxygen specifically? صح. So what? It will pull electrons in the covalent bond, which gives it a partial charge. صح. There are a couple of carboxylic acid derivatives which are amides and esters. Those formed from carboxylic acid. These are the biomolecules that you have to know. Amino acids, proteins. Say them with me. Sugars, carbohydrates. Fatty acids, ما في. ليش طيب ما في؟ ها؟ They are not a monomer. A lipid is what? A fatty acid plus a triacyl glyceride. So what? It's not a monomer of it. It's different. In the exam, I think, Jihad, I think this came last year. A fatty acid isn't, صح? إنه بالmidterm it came, okay? And then nucleotides form what? DNA, RNA. All of you in the back, you can hear me, صح? انتوا اللي ورا.
Please can you stay quiet just for the record, please.
What is the structure of amino acids? Amino acids, you have what? You have a central carbon, alpha carbon. Linked to that is what? Linked to that is what? A carboxyl group. To the other one is linked to what? An amino group. So, to link to the alpha carbon home بالنص, you have a R group. It changes from each amino acid. هلا amino acids are classified as they're all classified as either alpha, beta, or gamma amino acids. How are they classified? It's due to the what?
It's the position of the C, the carbon. Depends on which carbon the
It is the relation of the amino group with the carboxyl group center. بدك شو شيبس كده؟
No.
Attached to the alpha is the R group. R groups largely determine the function of the amino acid. They can either be hydrophilic or hydrophobic. تمام؟ هاي شرحناها. يلا يا اخوان. How many peptide bonds are shown above?
Chocolates, guys.
أحدا؟ يحيى. ها؟ غلط. صح. Can you point them out? This one? And? This one? صح. هلا. A peptide bond is a bond that forms between two amino acids. How? The amino group will bond with the carboxyl group, okay, via a nucleophilic substitution reaction. This is an amide linkage. ماشي? It connects two amino acids which forms peptides and polypeptides. How many amino acids are شو اسمه؟ peptides. غلط. صح. What mainly determines the three-dimensional structure of protein?
Hydrophobic interactions.
Hydrophobic interactions? لا. صح. ليش؟ Okay, hello. It's because of the interactions of the R groups. Some are hydrophilic, others are hydrophobic. If it is hydrophobic, inside the cell عندك. This is the protein. Outside of it is what? Or if it is شو اسمه? Water. فشو hydrophobic بكره المي. Hates water. Goes inside. And hydrophilic loves water. Goes on the outside the protein. And so what? It changes like the structure of the protein. What is the classification of the sugar above?
Come on, everyone, show of hands. You can make mistakes. It's okay.
Guys, make mistakes now, please. Embarrass yourself, make mistake عادي. Because in the exam, if you make a mistake, you will like ايش الفرق؟ آخر واحد بالزاوية. إيه؟ صح. آخر واحد. ما طيب. Pacademia حياكم فوز. هلا aldose شو هو aldose؟ الشوجر can be either A A sugar can either be a aldose or a ketose. Aldose shoe, it has an aldehyde functional group. C double bond OH. A ketose has a ketone functional group. ماشي؟ has a C double bond ORR. ماشي؟ Hexose shoe. 6 shoe carbon. 1 2 3 4 5 6. تمام. Give me an example of a aldopentose. انتوا شو لا واحدة منهم غلط الثاني رايبوز. طيب شو الكيتوبنتوز؟ an example of it. Example of like a sugar that's هات غلط صح. في هذا بنتوز؟ لا في واحد أنتو بنتوز. أنا آسف صراحة. أولدو بنتوز بصراحة كان صح. ديوكسي رايبوز حاجة ثانية. تمام. Fatty acids. Fatty acids are monocarboxylic acids with an even number of carbon atoms. What does monocarboxylic mean? One carboxylic group. Saturated أو unsaturated. Fat.
Saturated, no double bond. Unsaturated has at least one double bond.
صح. Or if it is a monosaturated, it has one carbon double bond. If it is poly unsaturated, it has more than one carbon double bond. This is a nucleotide, صح? What are the components of it? واحد. ارفع يديكم.
Show of hands.
تمام. كمل. صح. هلا the sugar can either be a ribose or a deoxyribose. ماشي. You can have more than one phosphate group. What is a compound that has more than one phosphate group that is a nucleotide? ايه الفايدة؟ تمام.
يا شوكولاتة الشغلة.
طبيعي. تمام. These are the bases. You have the bicyclic purines and the monocyclic pyrimidines. ماشي؟ شو عندنا البيورينز؟ adenine, guanine, pyrimidines, thymine, cytosine, and uracil. Which of the following molecules is not a component of nucleotides?
ليبيد.
صح. ايوه. اعطي اسمع مش صاروخ مش صاروخ جوال مش خليفه. طيب هذا شو DNA ماشي. DNA has a two anti-parallel polynucleotide structure that arranges into a double helix. اوكي شو طب نشرحها. Anti-parallel 5 to 3 عندك 5 end and then a 3 end. The other side anti-parallel عكسه 3 to 5 اقلبها بالشقلوب. 2 poly you have عندك واحدة and then 2 ماشي. What is the bond that links all of the nucleotides here? Nucleotides. Which are the bases? ايدين لو سمحتوا ايدين. اللي ورا. صح. ايه How is RNA different from DNA? ثلاث شغلات. RNA has a ribose sugar. DNA deoxyribose. It has uracil instead of thymine. السلام ورحمة الله. وهي أهم نقطة. RNA is a single polynucleotide strand. DNA is more. Jihad, دورك.
Thank you, Kasim. Thank you, Now, everyone, you're seeing the title of my lesson. Is the living cell a chemical factor? Someone answer. Yes, what? It's what? Independent. Any other answers? Do you know the scientific term? Why do we call it? That's correct, but I want the scientific term. Does anyone study? Autobiosis, correct. Autobiosis is based on three things: autonomy, self-organization, and self-maintaining. Can anyone just explain one of these terms for me? Anyone, or I'll be choosing at random? The guy in the pink. Yeah, what does autonomous mean? Self-organizing. Okay.
They organize products and proteins by their own.
Kasim said something in 1.1 that explains this. Can you recall it?
Not really.
Kasim, self-organizing. Which one of the 1.1 was it? Self-organizing.
قصدك من 1.1
Exactly. يلا. ان شاء الله تذكرتها؟ يلا احكي. Yes, wrong. Because that every single thing in your body starts as cells, then becomes tissues, organs. Don't you remember it? Hierarchy. That's what self-organizing means. Autonomous, that no one needs to tell you to do proteins, to burn lipids, all of these things. And self-maintaining, you're still alive. That's self-maintaining. Now, let's start with the main thing about this lesson. Biochemical reactions. A very important thing that you need to know is that we have a little or high amount of reactions in your body. The amount of reactions in your body, are they high or low? High. No, the types are low, but the amounts are extremely high. You have millions happening every single second, but the reactions are very simple. Let's get to it. Now, the first one. This, I have a little scar with it. It's one of my mistakes when I was in your age, so we're going to take it into detail. Here you have nucleophilic substitution reactions. They are composed of three things: nucleophile, electrophile, and a leaving group. Let's start. A nucleophile either is negative, an anion, or has a pair of non-bonding electrons, which here you can see it has a pair of non-bonding electrons. B, which was the electrophile. The word phile, does anyone know what is it? Loving. Do you know any other words with the word phile? No? Give him chocolate. Laugh, something, that was a joke. Now, electrophiles, they are deficient in electron density. They're deficient in electrons, so therefore they're more easily attacked by a nucleophile, which has a pair of non-bonding electrons. That's why they're easily attacked by it. Now X is the leaving group, which by leaving becomes a nucleophile by itself. It either becomes negative or it has a pair of non-bonding electrons. Hydrolysis. It's one of the main reactions of nucleophilic substitution reactions, in which oxygen of the water, oxygen itself, not the whole water, is the nucleophile. Can someone tell me why? Why is oxygen considered a nucleophile? Yellow.
Because it has electronegative, which means it loves proton.
That's correct, but what does it have? We said that it needs a criteria, one of the two criteria. What were they? Either an anion or a pair of non-bonding electrons.
And a lone pair.
Yes, a lone pair of electrons, yes. Now, when you take chapter 3, you're going to take the structure of water, and you're going to see the two pairs, probably with Kasim or Shazia.
The questions you bring here, is this the way they come in the quiz?
No, the questions right here, just to recall you, but when I bring you the multiple choice, that's the way they're going to come in the quiz. These questions were based on other questions that we had in the exam, and the main idea or the main point of the question was understanding this. So that's why I just put the understanding part, not the question. Now, we have elimination reactions. Elimination reactions, you start with one product, you end with more than one, and the most important thing, that there's a double bond forming. If there's no double bond forming, this is not an elimination reaction. The most common type is, can someone guess? Don't read it, someone guess. Dehydration, correct. It's when you remove water from an alcohol. Now addition, the complete opposite. You start with one of the substrate having a double bond, and it ends with removing the double bond, and two substrates end with one product. Now, we took two, here, like the other one was dehydration, here what do we have? Hydration, the complete opposite. We just add water. So, we took two reactions that involve water, hydration and hydrolysis. What's the difference between them? This is a hard question, you're going to get 2 candies.
Hydration is adding water to a compound. Hydrolysis is using water to break a bond.
So in hydrolysis you break a
You
bond.
break a bond.
But don't you break a bond in hydration?
When you
Isn't
use
it a
water,
double
when
bond
add
that you
water-
remove it?
When you add water.
So what's the critical difference?
Hydrolysis, the water isn't part of the compound. It's just some like an addition.
Can you get a round of applause? Now let's continue. Isomerization. Two. Yalla go on. Another one. Come on. You get it? خلاص. We kept the only 2. Isomerization. In isomerization reactions, there's just intramolecular shifts. Focus on this word. It comes in many questions. Intramolecular shifts that don't change anything about the substrate other than the position of the chemicals inside it. Intramolecular shifts. It's a word or a phrase that they use in the exams to refer to isomerization reactions. Keep that in mind. Now, redox reactions. Redox reactions happen between a reducing agent and oxidizing agent. What happens to the reducing agent? Oxidize. What happens to the oxidizing agent? Reduce. What is the main how can you see it, or without counting the electrons? How can you find out if this is reduced or oxidized? The book actually answers that for you. Can you tell me what it is?
Number of hydrogens and oxygens.
What about it? Does it increase? Does it decrease? With what? If it's oxidized, what happens? Less hydrogen, more oxygen. What happens in reduction? In reduction, right? What happens in reduction for the amount of oxygen and hydrogen? In reduction, less oxygen, more hydrogen. Now we're going to see examples about it. Which of these is more reduced? It's a very easy question. Someone, yalla. You, great. FDH2. Why?
Because it has more nitrogen.
Exactly. Now, this is a very important tip because in the exam, you have to expect a question showing you a reaction and asking you what type of reaction is this. So I just wrote my thinking process whenever I see a reaction for the first time in the exam. Yours could be different, yours could be the same. This is mine. First, I see, did dehydration happen? Or did 2 products turn into 1? No, this is... 2 products made from 1. Is it a dehydration reaction? Is it elimination? Did we get a double bond forming? If it's not, I look at hydration. If it's not, I look at hydrolysis. ATP hydrolysis. It's a very important example that they love to give you in your exams. Not only this, you're going to see it in the next term as well. Every single exam, keep it in mind. ATP hydrolysis, it's what kind of reaction? You. Yalla, someone answer. Nucleophilic substitution. You need to know this. It's very important. It's one of the examples written in your book. Then, if it's not like that, do I ask myself, did the chemical formula even change? If it didn't change, what reaction is it? Isomerization. Then, after I check all the other reactions, I look at the hardest one, redox, and count the numbers of oxygen and hydrogen. This is how I did it, and I'm in year 1, so I would suggest you to use it. Now, energy. Can someone tell me what energy is? Just read it. Someone answer.
Energy is defined as the capacity to do work that is to move matter. Cells generate most of their energy using redox reactions in which electrons are transferred from an oxidizable molecule to an electron deficient molecule.
Which kind of reactions did she say? Which kind of reactions did she say? Redox. Redox is the main reaction that we use to make or use energy. We use two molecules, mostly NAD plus and FAD. They take an hydride ion, which is an H minus ion, which is a hydrogen with a proton, a neutron, and 2 electrons. That's why it's negative. And here you can see that it becomes NADH and FADH2. Which is the oxidized one and which is the reduced one?
Reduced is FADH2.
And
Oxidized is NAD.
why?
Because the reduced has more hydrogen ions.
Exactly. Thank you. Now, there's nothing for me to say. It's just memorization, but it does come in the exam. You need to know the difference between autotrophs and heterotrophs, photosynthesis, chemosynthesis, all of these things. You just need to know them. Now, anabolic and catabolic. Expect many questions on this. Not only in here, also in the midterm, also in the final. They love to put questions about this part. Here, a question you might see. Which type of metabolic reactions or pathways use up energy? Catabolic, wrong. Anabolic, use. Exactly. They build up stuff so that could be cut by catabolic. Okay? Now, on the other hand, which makes energy? Catabolic, exactly. Okay, so as you can see here, this photo might seem very simple, but it's very important. As you can see, the interactions between catabolism and anabolism. Catabolism cuts down the energy or the food that you produce to give energy in the forms of what? What is the form of energy or the current of energy in our body? ATP, exactly. Another one, we just discussed it. NADH, another one. FADH2. These are high energy molecules that your body can use. But the currency or its ATP, as you're going to learn in chapter 4, that's coming in the midterm. Okay? So, catabolism creates more ATP, NADPH, and NADH. You're going to learn a lot about this in the second semester. You don't have to worry about it now. Now, these things that are more reduced, highly energetic, are used by anabolism, so you create the molecules you need to survive. Now, biological order. It's again just something you need to memorize, but you need to know that synthesis and degradation happens, transport of molecules, production, force of movement. These are the biological order of any organism. Now, final question, and I chose it a little hard. So, yalla, show me if you understood what I said. This is one of the hardest questions, and doctors do put this level of questions in your exams. Not a lot, maybe 1 or 2 per exam, but you need to be acquainted with such questions. If no one answers, I'm going to start to pick at random, so don't put yourself at the chopping block. Blue. Which one?
Happens at the same time. B, nucleotides.
Yalla, someone else, someone courageous. Exactly. What? Now, how can we solve such a question? Because most of these words are jumbled. How did you solve it? Why did you expect it? Exactly, that's step number 1. You need to read the question and to understand the question. And even if you don't understand the question, just pick keywords. They help a lot. He probably guessed.
No.
You didn't guess?
It says food, and food goes to the stomach, and the stomach contains acid.
Exactly. See, he took context clues. So at any moment you face a question in the exam. Even though it's filled of jargon, nucleophilic, electrophilic, acyl group transfer, they're just trying to confuse you. Just focus on the question, try to understand it, try to read it in a simpler way, and then you're going to see, what's your name?
Fuad.
Fuad, and you're going to answer just like Fuad did. Thank you everyone, that was me for today. Yalla, now we're going to take chapter 2 with Haya.
ارجع اعيد اسمي تاني. Chapter cells انا عارفه ان في كتير منكم لسه خارجين من البايو. By the way, is anyone here that doesn't understand English? I mean Arabic. English. Okay, I'm gonna try to reduce my Arabic words. We just came out of bio, and probably many of you didn't study. عادي, this chapter is very easy. Many of us took it in high school. There will be many new concepts, but if you pay attention with me here, it's going to be very easy. I know you're supposed to study from the book. The book is going to get into so many details, but here I tried to mention the main points. And before I start, I want to tell you, to give you an advice, because it happened to me in my last quizzes. In the book, you're going to see so many details.
I might check that.
Okay. الو اوكي كده حلو؟ اوكي طيب في الكتاب هيكون فيه many details. كتير منهم هيكون unnecessary and some of them are going to be very necessary. But what I want to mention is, and I want you guys to really focus on it. Don't focus on the unnecessary details and forget the whole picture of the main point. And I'm going to remind you command during the lesson here. Before we start, I want you to take you through the learning objectives so you know what's going on through our lesson. First of all, we're going to go through the basic structures of both eukaryotes and prokaryotes, and then we're going to go specifically through eukaryotes and prokaryotes with the distinguishing characteristics between both of them. طيب, as eukaryotes, as humans, we are complex organisms. We're made of millions of cells. I can't tell you in والله above 100 cells, والله above 200 cells, that we're made of billions of cells. We also know that cells are the smallest units of life. When we talk about cells, can you guys see? Okay. When we talk about cells, we have many different cells. We have endocrine cells, we have cardiac cells, we have nerve cells. There are differences between them, but they have the same structure. مثلا, a lot of our cells have the cytoplasm. All our cells have plasma membrane without exceptions. Some of our cells might have a nucleus, some of them might not have. Can you give me an example of a cell with no nucleus? Okay, can you guys not answer? No, our cells. As eukaryotes. Okay, can you answer your picture? No, I'm saying eukaryotes. Red blood cells, yes. Right. Why do red blood cells not have mitochondria? اسمعوا السؤال اللي قلت لكم. Here.
Red blood cells don't need to have energy for any reaction inside.
Not completely correct.
Because the blood cells it's the one that transports it. Doesn't need energy to perform its function.
Okay. Yeah.
It needs more space to play with.
That's also true, but it's the reason why we also don't have a nucleus. What about the mitochondria specifically? Can you guys please tone it down? I can hear you. No. طيب خلاص, do I answer it? طيب. Red blood cells, what do they transport? They transport oxygen. What does mitochondria do? It uses oxygen. I don't want my red blood cells to use the oxygen before I transport it to other cells. صح ولا لا؟ Because that was a side question بس عشان نفكركم. And now all cells, as I said, they're surrounded by plasma membranes. For all our plasma membranes, different cells, it doesn't matter, they're made up of the same type of particles, biomolecules. They lipids, they proteins, they carbohydrates. Like, here's another question for you guys. It's very easy. Can you read it? Do you think water is polar or non-polar, and how? And why do you think this property is even significant? I want to see new hands. يلا I'm saying the new people every day. I'm not saying new people. Thank. Okay. It's powder. طب why is it powder?
Because how much does it link up with other water there? It needs to be water should be hydrophilic, and this wasn't water with all separate from each other.
أيوة. Okay. Yes.
Because oxygen has a high electronegativity, it pulls the protons closer to it, so it creates a...
Exactly. So I think Kasim mentioned it also at the beginning of the lesson. Water is made up of oxygen and two hydrogens, and oxygen is electronegative. طيب, why is this property even significant? يلا عشان نخلص بسرعة. Yes. No. Yeah. The second question. Why is it significant? Why do I need my water to be polar, not non-polar?
So other water molecules can easily dissolve them?
Yes, that's true. هلا احنا our cells احنا we know like you know how they say in the water is the medium for all chemical reactions to occur. For a chemical reaction to occur, I need the particles to interact with each other. If my water is non-polar, معناها it's inert. I can't have something that's inert. I need it to interact with other molecules. خلاص. هلا now I want you guys to سيبوا كل الكلام ده على جنب, okay, and focus on the picture. How's the plasma membrane arranged? It's a bilayer. I have two layers. خلاص. If we focus on this individual part, I have the hydrophilic head and the hydrophobic tails. خلاص. Pay attention to how they are arranged because I'm going to ask you a question about this. I have two types of proteins. I have integral proteins and I have what? I have peripheral proteins. هلا on a side note, integral يعني ايه؟ يعني it spans the whole membrane من أولها لآخرها. I have the whole protein. Peripheral means إنه it's attached to a part of the membrane. Yeah. خلاص. Look again at the structure. I'm gonna ask you a question. Also on a side note here, this peripheral protein is attached by non-covalent bonding. Peripheral proteins that are peripheral proteins that are on the cytoplasmic phase. Here, look. هنا we have the outer, the extracellular matrix, out of the cell. خلاص, extracellular. Here we have inside the cell. This part, which is in the inside the cell, is attached by non-covalent bonding. It's important, by the way, so focus on it. طيب, why do you think the plasma membrane is arranged in this way? Again, I said إنه إيه? I'm gonna ask you a question. Why is it arranged in this way? Why not the opposite?
Because polar attacks polar, and the cytoskeleton and the extracellular matrices are both polar. So the heads rotate towards the water.
Yeah, that's true. We said that the water is the medium for all chemical reactions to occur. So for them to be able to like interact with water or stay in this watery environment, they need to have a hydrophilic part. Hydrophilic يعني water loving. خلاص. طيب, we're done with this. Throughout this chapter, you're going to see so many new concepts. مش so many, هم 4 حرفيا. We have self-assembly, we have molecular machines, macromolecular crowding, proteostasis. Guys, I want you in the exam to know you're learning so many new information. It's very likely and it's very normal for you to feel like, oh, what does this word mean? Wallah, this happened to me so many times in my biochemistry quizzes. If you look at the word and you know and you studied it, but you forgot it, يعني some of them are like self-explanatory. You can remember, you can recall what you learned from it. مثلا self-assembly يعني ايه؟ It arranges itself. طيب, macromolecular crowding زحمة. Molecular machines و proteostasis يعني it's very unlikely إن أنتم ممكن تفتكروا حاجة if you forgot anything about it. طيب, هلا for the self-assembly, I want you guys to look at this figure very carefully. Many structures in our body are supramolecular. Supramolecular يعني ايه؟ Look at the compound word. Molecular من molecules. Supra يعني ايه؟ يعني great. طيب, they are self-assembled. يعني many structures in our body are self-assembled. يعني ايه؟ Imagine إنه أنا I have puzzles. خلاص, I have a box of puzzles ورميته كده على this table. Okay, I'm very sorry. ما علينا. I'm speaking out of Arabic بس يعني ما علينا. Okay. I have puzzles. I threw them on the table. Suddenly, كلهم they came together. This is what happens in our body. خلاص? With the exception إنه supramolecular يعني great structures in our body. يعني I don't have a small molecule. I have a very great molecule in our body. It's very big, and it's self-assembled. طيب they arrange themselves together. كأنه imagine يعني شايفين هنا this part. شايفين how those two structures are coming together. زي الماجنت. كأنه self assembled. طيب هلا we can't let this process happen on its own. We need some kind of supervision. Who does this supervising for us? If you want to answer.
I have a question. Supramolecular, what does it mean? Like, it means big or?
Supramolecular means big. I have big structures in my body. حتى here I gave examples. These examples are very important. They're 100% asked about. مش 100% بس يعني they are very likely to ask about it. طيب I need some kind of supervision just in case. Sure. Ask. Yeah, molecular chaperones. Molecular chaperones are our supervisors here. خلاص؟ هلا for this supramolecular structure, you're gonna see so many bonds here. That's how they come together. These bonds are weak. خلاص؟ طيب, you're probably gonna ask, big molecules in our body, big structures, and they're held together weakly. How is this possible? When weak comes together, they become strong. عادي كأنه team, واحد ضعيف together. English? Yeah. Okay. And one person is weak and they're in a team, they'll become strong. خلاص? Okay. And now we're going to talk about molecular machines. I want you to look at the structure very carefully. Look at the cargo here. Look at those tiny legs. Well, this is the filament. This is the road upon which they move. And motor means what? And something is moving. صح ولا لا? أنا in a car. What moves the car? It's motor. طيب. إحنا عندنا cellular processes in our body. We have many multi-subunit complexes in our body. خلاص, many subunit complexes such as what? Kinesin methionine. I remember in the last semester, last year, they asked us about kinesin methionine, and it's an example of a molecular machine. خلاص, we have so many proteins, and part of this protein is going to be the motor protein. It's going to move the rest of the structure. طيب, I want you guys to see, and here, by the way, this statement is very important. I'm going to mention it here. How do they move? They use energy. And how do I move? I need to eat, صح ولا لا? Okay. They use energy. So this process is ATP driven. This is very important. خلاص. When they get this energy, they need to use it very carefully. Why? To move their legs in the right direction or orientation. This is very important. The right direction, towards the direction orientation is very important. Imagine مثلا in this leg here, instead of it moving like this, it moves like this. The cargo falls, صح ولا لا? طيب, if this happens in our cells, with some small mistake, very small mistake happens in our cell, خلاص we can die. يعني مثلا if the mitochondria doesn't do its job properly, what's going to happen? We're going to be deprived of oxygen. But it's very important in they move in the right direction and orientation. You guys have to know. The examples, when it's an energy-induced change in shape. خلاص, easy. طيب. Macromolecular crowding. Right now, there's صح في empty spaces here. صح ولا لا? مثلا there's a space between you guys. طيب, can I come right now and sit between you guys? No. Why?
There's no space.
Exactly. مع إنه there is a very tiny space I can squeeze between you guys, بس في what? في personal space. صح ولا لا? طيب, this is the same way for cells. Cells are composed of what? يلا tell me. Tell me, you guys are not focusing. والله. Tell me. لا, أنت you're always answering. Cells are composed of what? لا, like the very basic structure of cells. Atoms وإيه؟ Atoms جواها إيه؟ What are the subatomic particles? Electrons, صح. طيب, خلاص. We reach to the end point. We have electrons. You know how in high school they used to make us draw the electron clouds, the first shell, the second shell, whatever. These electron clouds, they will repulse each other between the molecules. That's why the molecules have a personal space. And as you can see here in this structure, these are empty spaces. Theoretically, if you look at it, I can squeeze a small molecule here. Okay? And I'm going to ask questions at the end of the lesson. It's going to be 4 or 5 questions. It's going to be exam-style questions. So focus with me. I can squeeze here. Theoretically, if I look, I can squeeze a small molecule here. صح? Okay. But in reality, I can't let this happen. Why? Because في steric repulsion or hindrance. The steric repulsion or hindrance is the atom's personal space. I can't just squeeze a small molecule here. This steric repulsion is also, it causes what? The excluded volume. And this volume, خلاص, just pretend it's not there, it's excluded. Need to know the percentages also, it's about 20 to 40%. طيب. I want to ask you guys a question before this. What's the difference between crowding and concentration? And if I say this room is very crowded, oh no, this room is very concentrated. Okay. Yeah.
Is that the crowding, there's irregular spaces between them, they're all bunched up into a small space and concentrated? It's like the regularly distributed throughout the scale.
No. Okay.
Concentrated is the same type of molecule in one space. Concentrated, the crowding could be
Exactly. Concentrated يعني ايه؟ يعني انه مثلا what's your name? يكتب. انه مثلا I have 100 يكتز in this room. This is what concentrated is. بس crowding انه لا I have يكتا سارة احمد محمد محمود خلص easy. شايفين these light circles هم دول ال excluded volume. Here مثلا ما بينهم I can squeeze a small molecule. خلاص. We're going to come to a very important concept, but it's also very easy. Signal transduction. When cells exist, they need to interact with each other. And in here, I need to talk to you guys. حتى if I'm not going to explain, at some point I'm going to have to interact with you guys. This is the same for cells. They need to interact. طيب, how do they interact? They have receptors, we have ligands. Ligands are what? Give me examples of ligands.
neurotransmitter,
acetylcholine, neurotransmitter. طيب صح. أنا have receptors. هلا what do they do؟ أنا عندي معلش نسيب البتاع ده ثانية. أنا هنا عندي receptor. عندي ligand. This could be a neurotransmitter, whatever, a hormone. It will bind. ده اسمه إيه؟ اسمه reception. زي عادي بدخل العمارة reception بروح للسكيورتي فين العمارة كده. This is the reception. طيب. The second thing is what? It's transduction. Transduction معناها إنه مثلا I have this is the receptor. I have the molecule. It fits like this, but it doesn't fit perfectly. Transduction معناها إيه؟ إنه receptor يضم إنه it comes along with the ligand. It fits with it. It undergoes a 3D conformational change. خلاص? We're done with reception. Transduction, we have the response. Response is very easy, يعني self-explanatory response. First of all, when the ligand binds, and when it first binds to the receptor, this is called what? The first messenger. خلاص? Now, I can't let the neurotransmitter go into the cell. I want it to stay outside. So basically what happens is the receptor tells the ligand, tell me what do you want to do, and I'll go inside and I'll tell them what you want to do. This is the response, and the first message is getting converted into the secondary message. طيب, once I get this message across, what's going to happen? So many changes are going to happen. There are going to be changes in gene expression. There are going to be changes in the enzymatic activities, in the cytoskeletal rearrangements, in the protein activation. طيب, I'm going to ask you guys a question. I don't know if you guys took this yet ولا لا. But how do I activate or deactivate a protein? No. A protein. عادي, an enzyme is a protein.
Genes.
Okay, what do genes do? How is this gonna happen? I'm gonna give you like a very small hint. It's about like covalent bonding. Some type of covalent bonding is gonna happen. What's gonna happen?
Hydrolysis.
لا, hydrolysis يعني I'm breaking it down. طيب. إيه؟ طيب. Phosphorylation and dephosphorylation is gonna happen. Phosphorylation can activate some proteins, or it can deactivate some proteins, or vice versa. طيب خلاص, now we come to the last step, which is about termination. And I have many ways that I can terminate the signal in. خلاص, I got the message across. I did the appropriate changes. I want to terminate it. How am I going to terminate it? Either and I remove the protein, which is the ligand, by destroying it. If it's a non-protein, I degrade it. Usually receptor, usually ligand, بيكون protein. If it's a non-protein, I degrade it, or I can deactivate it. I can keep it still in the receptor, but I can deactivate it. طيب. يلا. A question. What is our universal signaling device? Yeah, okay. صح. طيب. Do you think its concentration is usually kept low or high within the cell? sure. طيب raise your hand هما مش عارفين ايه؟ ها؟ No, it's usually kept low. It's usually kept low. When a change happens and our cells need to act upon that change, the endoplasmic reticulum is anyway, the endoplasmic reticulum is going to secrete this calcium and its concentration is going to be high. But before any change happens in our cell, the calcium concentration is low. طيب خلاص we're done with eukaryotes. Alhamdulillah. خلاص we have a small part left. We have prokaryotes. I want you guys to look at these two. For prokaryotes, I did this mistake in the first semester. There was a question that asked about the types of prokaryotes. It was bacteria and archaea. خلاص بس أنا because I focused on the unnecessary details in the book. I'm not saying all the details in the book are unnecessary because I focused too much on the small picture and I forgot the main picture. I was sitting على choices محتارة bacteria ولا archaea ولا whatever other choice there was. But don't do my mistake. The prokaryotes are bacteria or archaea. Whatever the differences the prokaryotes have, they still have the same basic structure. They have a cell wall. The cell wall is made of what? Can you guys raise your hand when you answer? But okay, it's peptidoglycans. And the prokaryotes, because they all have the basic feature: cell wall, plasma membrane, no membrane-bound organelles. No membrane-bound organelles. يعني any organelle that I have, mitochondria, nucleus, whatever, it does not have a membrane in it. خلاص, do you guys get it? No membrane-bound organelles. This is very important. And we have circular DNA. For the shapes of bacteria, you guys need to know I have 3 shapes of bacteria. They can be circular, Kokar. You can kind of Kokar, see, circle. Okay, well, I have bacular, which is cylindrical or rod shape. I have this other one, which is spiral shape. خلاص. Few special features of prokaryotes present in all prokaryotes. The first one is it has an ability to retain dye. يعني if I have bacteria here and I can see it with my naked eye, طبعا I can't see any bacteria with my naked eye. But if it happens and I can see it and I put the dye on it, it's gonna retain that dye. خلاص. It has a very small size. All prokaryotes are very small. Can you guys tell me what's the diameter of prokaryotes?
0.1.
It's 1 micrometer. Yeah. Okay. Well, they can move.
What do you mean by retain? What do you mean by retain?
Retain the dye. Do you know waterproof clothes? You know how if I spill water on a waterproof clothes, it's just gonna fall up? طيب, but if it's on a normal cloth, what's gonna happen? It's gonna absorb that water. The bacteria is the same. They can retain this dye, and they will not let it fall through or whatever. خلاص, understood? Clear? طيب. What determines the ability of the cell to retain specific dyes? Okay. مش موجودة الإجابة فمش هخبيها. What determines the ability of the cell to retain specific dyes? Can you answer? Answer. فيش غيرك.
That's about the archaea. The first time I ever heard of it, it's like the same as bacteria or like what is it?
يعني كلهم عائلات من عائلات البروكاريوتس. يعني you don't really need to actually understand what it is. Just know. لا, you don't have to go into the details. Just know I have 2 types of prokaryotes: bacteria وarchaea. خلاص. طيب. We come back to this question, guys. Please فكروا معايا. Just imagine I have a towel. What determines the ability of the towel to absorb water? Huh? What peptidoglycans? The cell wall? يعني the structure of the cell wall, yes, it's made of peptidoglycans. طيب. No, not really. طيب خلاص, it's the thickness. I tried to give you hints. Thickness. And if I have a thick fluid, it's gonna absorb more water, صح ولا لا? And if I have thin and I spill water and all the water is gonna fall down. خلاص, it's very easy, guys. In the exam, يعني it's gonna be the same way. You have to relate it to your own experiences or whatever. خلاص. هلا we said إنه cell walls are made of peptidoglycans. What are peptidoglycans? What does glycan mean? Sugar. Okay. Yeah. So I have so many sugars, and these sugars are linked by peptide bonds. A peptide bond is what? Huh?
The bond between amino acids.
Okay. صح. طيب. Okay. مش هدخل مش my lesson بس anyway. The outer membrane. لا شوفوا. هنا we have gram negative bacteria and we have gram positive bacteria. هلا this picture is very good لأنه it summarizes the whole thing. You can tell من the structures. We can tell here إنه the cell wall is made of peptidoglycan. هنا we have an outer layer which we're going to discuss in a bit. And between this outer layer and my cell wall there is the periplasmic space. خلاص? The periplasmic space is here. كمان the plasma membrane in prokaryotes is somewhat similar to our plasma membrane. يعني it's a lipid bilayer. بس أنا عايز بس أعطيك basic structure. طيب the outer membrane, which is the cell wall, is also made of something else. It's lipopolysaccharides. Do you guys know why when you get an infection, sometimes your fever can be high? يعني يلا ما حدش يقول ايه؟ يعني انت خربتي على انتين ما علينا بس اوكي يعني they release endotoxins these endotoxins by the way lipopolysaccharides act as endotoxins because they are one of the types of endotoxins that cause fever. طيب you have to know برضو انه the outer membrane is somewhat permeable. It will let substances in and it has pores. It has porins. طيب the same thing as pores. Okay. طيب what type of proteins are porins? Are they integral or peripheral? Integral صح. طيب هلا if I go back to this picture you can see the pink outer layer صح ولا لا? This is not the cell wall. It can be the slime capsule or the slime layer or the capsule. هلا look carefully at the picture. Look at how they are arranged. خلاص guys can you please stay quiet شوية؟ طيب خلاص هلا another name for the slime layer is a biofilm. I'm gonna give you guys an example of a biofilm that many of us might have saw. إنه when someone has tooth decay or plaque, don't you see it's kind of like a yellowish layer? This is the slime layer or the biofilm. So the biofilm is another name for the slime layer. طيب هم كلهم يعني it's an outer layers. They both have different functions. For example, if we talk about capsules are like a mechanism for bacteria or prokaryotes in general to protect themselves from our immune cells. How do our immune system realizes if there is like a foreign substance or a foreign cell in our body? We have some type of thing on our membranes. Nitrogen? No. It's kind of true. But what in the glycocalyx? I answer خلاص?
Antigens?
No, مش antigens. لا. What differentiates our self cells from the non-self cells? Carbohydrates. Okay. صح, I have carbohydrates. I have self-carbohydrates. With those bacteria or archaea, they have what? They have different carbohydrates than our carbohydrates. فإحنا we distinguish. Our body knows our carbohydrates.
What's that carbohydrates?
طيب. What's the difference between both? Look at them structure-wise from the picture. What can you differentiate ما بينهم? Huh? طب slime layer what? Yeah, also صح. True. الslime layer it's irregular. صح ولا لا? Capsule is perfect. Perfect. But the slime layer is irregular. طيب. What about the function?
Slime layer reduces the loss of moisture, stops dehydration of the cell.
Yeah, true. With the capsule avoids what? Avoids detection. Do you guys hear his answer or no?
No.
Okay. Can you say it in a louder voice?
Okay.
The slime layer.
The slime layer stops dehydration of the cell. It stops losing moisture.
Yeah, true. طيب خلاص. I told you guys that the bacterial plasma membrane is somewhat similar to our plasma membranes, but there is one major difference. It's something that we don't have in our plasma membrane. Can you point it out? Okay, Okay, but we're talking about the plasma membrane itself. There is a major difference in the bacterial plasma membrane and in our own plasma membrane. What is it? It's here in this picture. Answer. Okay, it's a difference, but it's like a minor difference. في حاجة جوه. أنا بديكم هنتهي جوه. هوبانويد. خلاص. طيب هلا in fact احنا in our body we have something similar to the hopanoid, which is cholesterol. Cholesterol وhopanoid, they have one function, they stabilize our membranes. خلاص, you have to know this, they stabilize our membranes. طيب when we talk about the cytoplasm of prokaryotes, in the cytoplasm there are specialized regions, there are functional compartments. What are these functional compartments? They are specialized regions that have special functions. And one of them is the nucleoid region. In the nucleoid region, you're going to find two things. من اسمها, من nucleus. What does the nucleus have? Chromosomes. Type, exactly. And in prokaryotes, I also have chromosomes. But I also have another thing, which is the plasmid. طيب, اسمعوا. There's a difference between the plasmid and the chromosomes. الاثنين DNA. بس yeah, true. But also function-wise. Can you answer it? Okay. Our chromosomes carry vital genes. Vital genes إنه مثلا في الprokaryotes. مثلا they carry how genes and how the prokaryote will look like, the length of its pili, of its flagella, whatever. لكن in plasmids they have unnecessary genes, thus they give the cell an advantage. Like what? Let's say a cell got exposed to like a different مثلا إحنا we don't have plasmids. Okay. Prokaryotes, they got exposed to some other cell that's gonna attack them. خلاص? But they're gonna have what? They're gonna have bacteria-resistant genes. إحنا in the plasmid, the bacteria-resistant gene is not an essential gene. But it's an advantage. We have inclusion bodies. Inclusion bodies, يعني it's a dot. فيها a substance. It can be an organic substance or an inorganic substance. What is an organic substance? Yeah, it has carbon hydrogen bonds. طيب. خلاص, it has. Okay, it has carbon hydrogen bonds. These organic substances can contain phosphates or sulfates. طيب, why do I need phosphates? I mentioned it earlier in this lesson, by the way. Why do I need phosphates? Yeah, answer. For energy. Kind of true. This is not the answer I'm looking for.
Phosphorylation.
Yeah, phosphorylation, indeed phosphorylation. And I also need it to synthesize DNA. خلاص? DNA is what? It's a nucleic acid. And I have ribosomes. The points I mentioned here are very important. Please يعني go through it before the exam. طيب. Okay, when I look at the structure of prokaryotes, I have we mentioned the DNA in the nucleoid region, and then I have the flagella, and I have the capsule, and we have the pili. The flagella is responsible for what? Locomotion. خلاص. I use flagella to move. The prokaryotes use flagella to move. And pili, they have يعني a good amount of functions. I have one type of pili called sex pili. Basically, what it's gonna do? مثلا, our prokaryotes are gonna attack our body. They're gonna meet our cells. إحنا الhost. Okay? They're gonna meet our host cells, and they're gonna want to attach to our host cells so they can take over us or exchange DNA. خلاص? Did you guys know when bacteria attack us, when viruses attack us, they use our machinery to like survive inside our body? Yeah. خلاص, okay. فهنا prokaryotes, the pili, this is called the sex pili, perform a process called conjugation. Conjugation means transferring genetic information between the host and the prokaryote. Pili can also attach, it's going to attach to food sources or our cells. خلاص, eukaryotic cells, we came to the easiest part. I'm going to ask you guys a question here. We'll try to think carefully because this is not an easy question. But it's also easy. المهم, why do our eukaryotic cells have membrane-bound organelles while prokaryotes do not have membrane-bound organelles? Okay.
Surface area?
Yes, صح. I needed to increase the surface area because prokaryotes are small. They don't need this much energy. But we're complex organisms, we need so much energy to survive. So we need a high surface area for more chemical reactions to occur. Also, a point I forgot to mention is, we're going to go back for a little bit, macromolecular crowding. Why is this concept important? If I have more macromolecular crowding, this is a good thing. Why? Because cells are going to be closer together. When they're closer together, or when the atoms are closer together, what is more likely to happen? No. What? When I have macromolecular crowding, why is it a good thing? Yes, answer.
Molecules are more likely to interact.
صح. The chemical reactions are going to occur faster. طيب هلا in eukaryotic cells our organelles are individually regulated. خلاص I regulate the mitochondria on like its own, and then the nucleus on its own, and everything on its own. And the key for any chemical reaction to occur is a high enzymatic concentration. If I don't have enzymes, the chemical reactions are going to proceed very slow, and we might die. We need quick. يعني أنا speed up. أنا speed up ولا? Okay. Okay. بعدها outside the cell, outside our cells. يعني if anyone is talking يعني please خلوني أشرح. Outside the cells, what do we have? We have a glycocalyx. Glyco يعني إيه؟ Sugar. طيب صح. Outside our cells we have a glycocalyx. The glycocalyx is عبارة عن polysaccharides and glycoproteins. So outside our cells we have two things. We have the glycocalyx and we have the extracellular matrix. The extracellular matrix is produced by what? By fibroblasts. How do fibroblasts produce the extracellular matrix? They produce structural proteins and carbohydrates. This is very easy. The rules of carbohydrate molecules. We said cell-to-cell recognition. If my cell wants to recognize another cell, they need to have what? They need to have carbohydrates. And for adhesion, and for identity molecules, and receptor specificity. Our receptors are made of carbohydrates, by the way. And ligands are made of what? Ligands are made of what? Proteins. ركزوا. Okay. Okay. But the rules of the by the way, guys, we're about to finish. The rules of the extracellular matrix is to bind cells together. It has a supportive and protective function, and it regulates the cell behavior. Yalla, question. How does the extracellular matrix regulate the cell behavior? I'm going to remind you of something. The extracellular matrix has structural proteins, and it has carbohydrates. But how does it regulate the cell behavior? No, not really. Yeah, it does have cell adhesive molecules the same. How is this going to regulate? They're going to be closer together, but it's not going to regulate them. ممكن من جوه the whole process يفرق. How do you know? طيب. We have structural proteins. We said proteins are ligands. Ligands بيعملوا ايه؟ They transmit messages across the cell. طيب. هلا finally. How do our cells stay in place? How do they not fall off? Will they become curvy? Will they irregular? They have what? They have a membrane skeleton. The skeleton is attached. The skeleton is a point بيدي من inner membrane. خلاص? So it's attached to the proteins via non-covalent bonding. You also have to know what the membrane skeleton is made of. It's made of actin and spectrin and actin binding proteins. خلاص? This is very important. طيب what are the rules of the rough endoplasmic reticulum وسموث endoplasmic reticulum وما حدش يجاوب من غير ما يرفع ايده. يلا حد جديد.
New hands, guys.
New hands. حد. Okay, there's another rule for the smooth endoplasmic reticulum other than synthesizing lipids. What is it? Calcium storage. صح. الو جواد اديها شمس والله جاوبت. حلو حلو اللبس اسود يا عم. كله لبس اسود. اوكي. ما علينا. خلص. There is a special role for the smooth endoplasmic reticulum of hepatocytes. What are hepatocytes? Yalla fast. Don't raise your hand. Answer. Liver cells. They produce what? VLDLs. VLDLs are very low density lipoproteins. The last thing, before the last thing, We have the endoplasmic reticulum stress. What does it mean? Our rough endoplasmic reticulum synthesizes proteins. Now this complex process, it can go wrong sometimes. We can have a misfolded protein, we can have a protein that's not unfolded, that's not folded properly, or it's just unfolded. The failures are going to happen. So what are the reasons for this failure? There could be an activated inflammation process. Okay, it could be due to genetic factors. It could be due to environmental factors. There's infection, there's injury, there's illness. Are our body, is it going to allow this to happen for a misfolded protein to continue and carry out our processes? No. There are different types of stress. If anything goes wrong, we call it a stress. There are different types of stress. The minimal stress, okay, and then there is the severe stress. If the stress is minimal, a process is going to come into place and it's going to help. It's called the Eyad. It stands for what? Can anyone tell me?
Misfolded protein.
Eyad. اختصار لإيه؟ Endoplasmic reticulum associated degradation. Okay? It's going to transport, it's going to take the misfolded proteins, it's going to transport them into the cytoplasm, and they're going to be destroyed by proteosomes. If the stress is severe, two things could happen. The rough endoplasmic reticulum initiates the UPR, which is the unfolded protein response, to restore proteostasis. Proteostasis is similar to a word we took in bio. Can you relate this word? Yeah, exactly. Homeostasis. Homeostasis يعني I have a stable internal environment regardless of changes in the external environment. This is the same for proteostasis. I need to have a stable set of proteins in my body. By the way, the set of proteins we have in our body is called a proteome. Okay? A proteome, we all have different proteomes. No person has the same proteome as the other person. And in us, it changes every second. If I go into the heat, it's going to change to cope and adapt to the environmental conditions. We go back. If the stress is severe, the rough endoplasmic reticulum will initiate the unfolded protein response. What is the unfolded protein response is going to do? It's going to be like خلاص into the nucleus. It keeps on producing misfolded proteins. Stop it. Don't produce proteins. So it's going to stop protein production by the nucleus, and the nucleus gives the signals. And then another process will happen if the stress is severe. Autophagy could occur. Autophagy, I got the damaged organelle and I completely destroy it. And if I do all of this and there are still misfolded proteins, there is only one solution to this. We kill the whole cell. Apoptosis is going to occur. خلاص? Now for the last thing, when I produce proteins in the rough endoplasmic reticulum, before they leave the cell by exocytosis, they need to go to the Golgi apparatus. The Golgi apparatus is going to modify it. It's going to process the protein. Process يعني إيه؟ يعني it's going to see what type of proteins it is. It's going to modify it. How is it going to modify it? It's going to modify it by glycosylation. Glycosylation means what? Adding sugars. And it's going to package it and then it's going to send it outside of the cell by exocytosis. We have two sides for the Golgi apparatus. We have the cis side and the trans side. The cis side of the Golgi apparatus, this is the Golgi. The cis side is the one that's closer to the rough endoplasmic reticulum. And the trans one is the one that's closer to the plasma membrane. Do you guys know what exocytosis is? صح ولا لا? Okay. Why does exocytosis occur at the trans side and not the cis side? Why does exocytosis occur at the trans side, not the cis side of the Golgi apparatus? We're looking. Yeah, it's closer to the plasma membrane, فعشان بتطلع بسرعة. And by this, خلاص we reach to the end of our questions. Do I have time? Hello, Shazia. Do I have time? Or no?
We have half an hour.
For you?
For the entire chapter. Next chapter.
For the next chapter. طب خلاص. I'll ask the questions later on in the group. وخلاص. That's it.
يا جماعة رجعت لكم يعني round 2 يلا 3.1 molecular structure of water. Now I will be going a bit quickly because we are a bit short on time now. Okay. Now, what is the molecular geometry of water molecule? صح. صفولة. Why is water a polar molecule? هات.
Oxygen is more negative.
صح. تمام. Structure of water. شو قلنا؟ Water has a band geometry. Oxygen is more electronegative than hydrogen, so شو it will attract the bonded electrons in the covalent bond a bit closer to the oxygen, and that gives oxygen a slightly positive charge and gives hydrogen gives oxygen a slightly negative charge and gives hydrogen a slightly positive charge. Water is a dipole molecule because of that. Non-covalent bonding. What are salt bridges in proteins? D. We said what are salt bridges in protein? Interactions between oppositely charged amino acid side chains. Why do salt bridges جاوب هات
B. Hydration fines.
صح. هلا salt bridges fall under ionic interaction. Okay? Ionic interactions occur between oppositely charged ions. Sodium positive, chloride negative. They also form salt bridges. Salt bridges are formed a lot in the presence of water. As we said, hydration is of preferred. مش دكتور. بكير. دول.
Do you have to memorize common salts?
Common شو؟
Salt?
Salt? لا بس إنه because it's called inside the protein it's called a salt bridge. لأنه شو you have two R chains with opposite charges. It's called salt أتوقع because you know sodium chloride شو بيطلع عندك؟ شو اسمه؟ table salt. What best describes a hydrogen bond? سؤال what best describes hydrogen bond of these? B. When a hydrogen nucleus is attracted to the lone pair of electrons of an oxygen or nitrogen atom in a neighboring molecule. صح. Why are hydrogen bonds weaker than covalent bonds? D. Hydrogen bonds have a longer bond length. صح. تمام. Hydrogen bonds, as we said, the electron deficient hydrogen is weakly attracted to the unshared pair of lone electrons on an oxygen or nitrogen or a fluoride atom. The attraction is due to the large difference of what electronegativity between the two atoms. How many hydrogen bonds can water form? I have maktuba, 4. And hydrogen bonds I said are weaker than covalent bonds. What type of force arises when a biomolecule contains a neutral permanent dipole approach each other? Van der Waals, صح. هذا السؤال؟ طيب. Van der Waals forces. These are the weakest of the ionic interactions. They are the weakest of the forces. Okay? شو هن؟ They are weak electrostatic interactions between different biomolecules. These molecules can either be neutral, permanently polar, or induced polar. You have 3 types. Dipole-dipole interactions, dipole-induced dipole interactions, and induced dipole interactions. These are the three types. A dipole. A dipole issue. The best example is what? A water molecule. What? It has a charge on the hydrogen and a charge on the oxygen. It has a permanent partial charge on them. So, what happens? A water molecule, or this one, or it's like that molecule, will be attracted to the partial positive charge of the other one. So what the partial negative will be attracted to the partial positive of the other molecule. Dipole, induced dipole, you have a permanent dipole molecule. It will distort the electrons in a different molecule, okay, giving it a partial positive charge. So what it will be interacted to the other molecule. Induced dipole. You have two neutral compounds. Sorry. Okay. Sometimes there's a small distortion of the cloud, of the electron cloud. This will form a partial positive or a negative charge. So what it will induce, again induce a partial charge in the other molecule. So what it becomes? So it becomes induced double reaction. Now Shazia will explain 3.5.
Hi guys. Just kidding. That was just for the sake of entertainment. Anyway, so I'm gonna present 3.4 to you guys. And long story short, this entire part is about how water interacts with other entities. Now, solvation spheres, how it interacts with ions. Structured water, how it interacts with macromolecules. Dielectric constant, how it acts as a solvent, and so on. So I'll just go over it as quick as possible so I don't take up too much of your time. Alright, so we know, I think most of my colleagues have covered this concept already. Water is the universal solvent because it is polar, yes. So it can form hydrogen bonds. What kinds of atoms make it polar? Or like what kinds of atoms are electronegative that make it polar? Oxygen. Sorry? Okay. Anyone? Any other elements? Nitrogen. And fluorine. Yes, FON. So how much a compound or solute dissolves in water depends on how much hydrogen bonding occurs in the solution with water. So these are three organic compounds. Organic compounds are generally what? Non-polar. But these three in particular have a functional group that makes them polar and soluble. And that's alcohols, aldehydes, ketones. My colleagues have explained them, I'm not going to go through them again. Alcohol specifically is a type of group that can form hydrogen bonds with itself. It doesn't need to be dissolved in water. Okay? So, yeah, that's the point. Question? Oh, yes.
Is sulfur one of the compounds that can form hydrogen bonds?
It's an element, and if you find it in a molecule, like, I think you'll have a sample question. So in a particular molecule, if you find sulfur with hydrogen, it can form hydrogen bonds. Yes. Okay? Sorry, no, not sulfur. Fluorine, oxygen, nitrogen, not sulfur. No. Alright? So, question. How many hydrogen bonds can one water molecule form? Four. Two of them are through hydrogen, and two of them are through electron pairs on the oxygen itself. Okay, question. Who wants to answer? Yes? D? It increases? Any other answers? Decreases. Who says decreases? What happens to the attractive force between ions when you put them in water? Say you're dissolving salt in water. What happens between sodium and the chloride ions? Decreases. Right? That is the correct answer. So, do we know why it decreases? Yes. So, okay, guys. The dissolution of a solute in water occurs because it dissociates. Why does it dissociate? Because water molecules attract those individual ions more than they attract each other. So you have, for example, in the next slide, so a solvation sphere is formed when these water molecules are attracted to the ions individually, not sodium chloride as it is. So water comes in between and it attracts each of them individually, and sodium and chloride are like separated, right? So that is what a solvation sphere is. When it's formed fully, like throughout with all the solutes, full dissolution occurs. Otherwise, in another case, which I will show, so this is sodium chloride, right? Full dissolution. What happens if you have an organic compound like this? Crazy. It does not fully dissolve. Why does it not fully dissolve? Any guesses?
Because the interactions between molecules are stronger than the interactions between water molecule and the
Yes. And that the interaction between these two ions are stronger than the interaction between water and this ion. Why? Because they're covalent. Sorry. These are covalently bond, so they're stronger. So they're difficult to separate, which is why dissolution occurs less in organic compounds or less ionic compounds, covalent compounds. Okay, what does the dielectric constant of a solvent indicate? Its ability to, the ability of the solvent to dissolve a hydrophobic molecule. Yes. Is it A? A. Any other guesses? Yes. D. This one?
Yes, D.
Capacity of a solvent to reduce electrostatic attraction. Okay. Any other guesses? Yes. Yeah.
C.
C. Temperature at which the solvent boils. Interesting. Okay, so we're having a mix of A, C, and D. So the answer is going to be D. Why? It's pretty obvious with the name, isn't it? Dielectric. You're going to have dielectric constant is the name given for the ability of a solvent to dissolve solutes. So far we spoke about how a solute is dissolved in solvents. Here it's about the solvent's ability to dissolve. So that's what it is, right? Do you guys follow me? Are you getting what I mean? So when an electrostatic interaction between, say, like our previous example, sodium chloride, right? When it's interrupted by the water molecules, they're dissolving. So water has a high or low dielectric constant? High. Yes, correct. It's the universal solvent after all. Okay. What is the state of most water molecules in living organisms? So if you guys haven't noticed, by the way, I'm asking questions about topics I haven't mentioned yet, so I can figure out. Yes, again.
Non-covalently associated with membrane?
Correct. Any other ideas? Anyone's thinking any other option? Nobody? Okay, not bad. So it is correct. Non-covalently associated with membranes. Do we know what this is called? This association of water with macromolecules. Yes. Adhesion. Not really.
Structured water?
Yes, structured water. Who said that? Yes, somebody give them candy, please. Yes, it's called structured water. So, long story short, it's basically the same as a solvation sphere, except what's the difference? In solvation sphere, you had the ion. In structured water, you have a macromolecule, or molecules on the surface of a cell. So they also have charges, right? Lipids, carbohydrates, charges, so they attract water. Now, say for example, the core of your body is an oxygen atom. Your arms are 2 electron pairs. Your legs are 2 hydrogen atoms. So you can form 4 hydrogen bonds ideally, and one of them is non-covalently attracted to surface, and the other 3 are forming with other water molecules. So you just keep forming this whole network, and it's called structured water. It's a network of these water molecules in a 3-dimensional structure. So it forms like a shell, basically, pretty much, right? And what does the shell do? It protects, it separates, and since it's water that's forming the shell, it's going to be giving it, there's flexibility and structure, okay? We ignore that, anyway. the relationship between the motion of the structure of water molecules and their proximity to polar surfaces? I want you guys to take your time to think about this. Is that a doubt or an answer? See, damn, okay. Closer water molecules have restricted motion. Did you guys see this before? Oh. Right, you did. Okay, so I want you guys to think about it. Would any of you say the answer is D? Like, if you saw this in an exam? No? What if you interpreted it as water molecules closer to the surface? Okay, my point is, you can overthink like that sometimes, and you need to avoid that, especially in biochemistry, right? When you see a question, I want you to first think of what the potential answer could be in your head, then go through the options, okay? And, alright. Another question. Who wants to answer? You guys can, just raise your hand, read out the question, and answer it yourselves. The one to the left, please. E. Stabilization. Okay, the question. Can you read it out for us?
I can't see from here.
Dog. Okay. Although van der Waals forces are small, hydrophobic substances form cohesive droplets in aqueous solutions. What is this? Like oil and water, right? It's a specific example of hydrophobic attraction. So you're saying E is the answer.
Yes.
Stabilization of the hydrophobic phase by van der Waals interaction between water molecules.
Up to the left.
C.
I don't know. What do you think?
I don't know. I didn't see.
Dog. Okay. C. Someone said C. Hydrogen bonding between water molecules forming cages around the non-polar droplets. That is the correct answer. Bingo. So why is it not E? Any guesses?
Because it's a random guess.
Why are we random guessing? I want to test you guys on it. Yes. We'll give him a test.
Because water doesn't form van der Waals forces.
Water?
Doesn't form van der Waals forces.
Doesn't form?
Van der Waals forces.
Doesn't have what van der Waals forces between. Well, technically it's not, it doesn't. But the primary forces between water is what? Hydrogen bonding. What about within a water molecule? Yes, correct. Yeah, my bad. So where is van der Waals coming into play? Inside? Yeah. See? The hydrophobic structure, right? Hydrophobic molecules are attracted by van der Waals forces. And this is basically, long story short, the hydrophobic effect. Another question. What are the resulting structures when hydrophobic substance is added to water? And when amphipathic substance is added to water? So you have two cases here. Any new hands, guys? Anybody who wants candy and hasn't gotten it yet? Nobody? Okay. Yes. B. Micelle and clatrate. So you're saying you add hydrophobic substance to water, it forms micelle. And when you add amphipathic to water, it forms clatrate. Any other options? Yes.
What does micelle mean?
Okay, so you don't know yet. Alright, we'll address that. D. Clatrate and then micelle. So you're saying the opposite. Okay. A. Okay, so we're having a huge mix of answers.
I'm saying C.
So basically everything, right? A, B, C, D. Okay, yeah, you're welcome. I made this question very confusing on purpose. So the answer is clatrate
She answered.
being formed. Yeah, she did. Yes. We can give her candy. So clatrate is the structure formed when you think of oil in water. That layer or the droplets formed is what you call a clatrate. Okay? And what is a micelle then? What is amphipathic molecule? Am I going too fast? Do you want me to address the whole thing?
Can you explain
Yes.
what the words mean?
Here we go. Yeah. Oh my. Okay. The content has been mixed up. Okay. So here you see a micelle versus a bilayer. Okay. Firstly, yeah. So what does this look like? Any guesses? Yes. This is the pictorial representation of the hydrophobic effect. So you have completely non-polar, completely hydrophobic molecules in water, and they are hydrophobic. They want to avoid as much interaction as they can with the surrounding water molecules. So they form this clustered structure, and it's called what? Clatrate. Clatrate, hydrophobic. Okay? And there's two things that stabilize this formation. One is the formation of van der Waals interactions between the hydrophobic molecules. And two is it's hydrophobic, right? So what will make you happy if you have a phobia of something? Staying away from it, right? So So minimized surface area contact with the surrounding water molecules makes it more stable. So those are two factors. The text has been mixed up. Apologies for that. Anyway.
So the answer is which one?
B. Yes. Your question?
Your question, hi. This
Sorry?
is the water cage.
Water?
You said water cage like a slider joke, but is this the water cage like a slider in your basket?
Water what? I'm sorry.
You said when non-polar molecules
Yes.
enter water, it's like a water cage or something.
Yes, that cage is not this. This is not the cage. This is called a micelle. Okay. So this is the cage around a hydrophobic molecule. By caging, I just mean water likes itself a lot, right? There's so many hydrogen bonds. So they're bonding with themselves. And that is the main thing that pushes the formation of a clathrin. I want you guys to know this, right? I think we had a question like this similar. Guys, right? And what is a micelle now? So hydrophobic molecules have zero polarity, right? Amphipathic molecules have a polar head and a non-polar tail, or a polar region and non-polar region. So a part of it is attracted to water. The other part is not attracted to water. So what's going to happen? In hydrophobic effect, you're just minimizing surface area. There's no choice, but you're giving contact between hydrophobic and hydrophilic molecules. But in micelle, you see these round structures. What does this remind you guys of? Anything? Lipid bilayer, right? Yes. So it's similar to that. You can assume this is the phosphate head. So that's in contact with the water, because why? Hydrophilic. Okay? And the tail is hydrophobic, so it's minimizing its contact with water. So this structure is called a micelle. Are we following? Any doubts? All clear? Now, back to this question. Do you think any other option could also be right? No? B. No? See, okay, let me clarify this. Hydrophobic can only form clathrates. Clathrates I showed in the other picture, right? Amphipathic generally forms micelles. Now, so A, yes, that's what I was getting at. So A can also be correct, right? What changes? What's the difference? I just did this to confuse you guys to see if you were aware of the difference. These are formed by the same molecules. And what are these molecules? Amphipathic molecules. What changes? What's making them arrange themselves into micelles versus bilayers? That's just a circumstance, right? So when they generally ask you a question like this, they won't give you two options like this, but just like that. Yes?
So you have to know what amphipathic substances are, because like...
No, you don't need to know examples, but have you understood what amphipathic molecule is?
And they basically form micelle or bilayer.
Yes, but what in terms of a chemical molecule, what is it?
It has a hydrophilic head.
So fatty acids are hydrophobic molecules, so?
Yeah.
So when it ionizes, when it loses a proton, this area develops a charge? Yeah? You guys follow me? So when it has charge, what does it do? Become hydrophilic. It's attracting water. Yeah? So that part is polar now. But this part remains non-polar. There's a long chain of carbons. Okay? So that makes it an amphipathic molecule. You just need to know that particular detail. Okay? One last question. One more question. The plasma membrane is which of the following? A. What's a dialyzing membrane?
No, it's more than one option. It's also yeah.
So how many options is it? 3. Who said 3?
I don't know.
Yes. Fair enough. Yeah. Okay. So what is a dialyzing membrane? We'll get to it. So a plasma membrane is selectively permeable. We agree?
Yes.
It is an impermeable membrane? No. It allows things to pass through it, right? A dialyzing membrane. What's dialysis? You're passing blood through a tube. You're letting water move in, clean fluid move in. You're letting waste solutes leave. So you're letting the movement of both what? Solute and solvent happen. So that is what the dialyzing membrane is. And that is also the same thing as D, which I wanted to test you guys on. Alright? So this is osmosis in a nutshell. It's the passage of only solvent. there is no passage of solutes. But in our cells, we have dialysis. We have a dialyzing membrane, so it allows both to pass. But in osmosis in general, it allows only solvents to move. I'll make this clearer with the next example. And the membrane you use is called a semi-permeable membrane. That is what allows only solvent to pass. Allowing the solvent to move? Okay, so I want you guys to choose. Water or solvent moves from region of what to what? When you have something like this, right? The circles are your solutes. Sorry? Of what though? Solute. High to low solute. Water moves from high to low solute. Okay, so this is high solute concentration.
Solute moves from high to low. I don't know.
Solute doesn't move, yani. Solvent moves across the membrane. That's the tricky part. Yes. Oh, she said the same thing? Okay. Okay, to break it down. You have high or low concentration of solute here. Guys, please follow me. Bear with me. It's like another 5 step, okay? This is an area of high or low solute concentration? High. And solvent concentration is relatively lower, okay? So which way will only the solvent move? Left to right or right to left? Left to right. So you're gonna equalize the ratio of the solvent and solute molecules, okay? If you imagine this in your head when you're thinking about hypotonic, you don't need to worry about this ever again. You don't need to touch the topic. This is as good as done, okay? So, water or solvent moves from left to right, okay? So it's moving from region of concentration in terms of solute.
Low to high.
Low concentration of solute to high concentration of solute. In other words, also solute concentration is the osmotic pressure, okay? I don't want you guys to confuse yourself. They'll say high solvent concentration. And would that be high or low osmotic pressure? Low. Low osmotic pressure. Good job. Okay? So that's one option, right? Low to high solvent concentration. No, high to low solvent concentration. And low to high? Solute concentration, also known as osmotic pressure. Okay? Good job. Any doubts? Any confusions? Yes?
Can you repeat the word ammonia?
Okay. More solvent, less solute, less osmotic pressure. Okay? Less solute equals less osmotic pressure. So which way will solvent move? Low concentration of its solvent. But from? Yes, sorry. Thank you. Low concentration of solute to high concentration of solute. High concentration of solvent to low concentration of solvent. You're going to equalize it. Yes. Don't put it into words again. Just image, create the image in your head and then put it into words. Okay? If you get that down, you don't need to worry about it. It sounds really complicated, but it's okay. Alright. One last thing regarding osmotic pressure. So, does anyone understand this definition? Or we can skip past it if you guys do. The force necessary to resist movement of water across a semi-permeable membrane. Why are we resisting? Alright. Any? Nothing? Okay. So you have a column of water here. A column of a solution, and you have a beaker of another solution. 1 and 2. Alright? So if you see a solution this high, you'd expect it to go down, right? Physics and whatnot. It's not going to stay this way. That's it. Right? So why is this not moving? Let's say one being lighter is, guys, please, another few more minutes, okay? 1 is a solution of salt water. 2 is a solution of more concentrated salt water. So what will happen? Which way will the water move?
1 to 2.
1 to 2. So it's going to go this way, right? It's going to increase the height of the water column. And what is this? It's the semi-permeable or osmotic membrane. So it's going to allow only fluid to pass through. And fluid is going this way. So there's a force that's pushing this down, right? There's no way water is going to stay at such a high column. But what is resisting it? This osmotic pressure, because they are now equal, right? The water has moved in. It has created the same concentration of salt here as it has here. Any confusion? No. Good? Perfect. So, question. you guys know what RBCs are isotonic? RBCs, red blood cells. Oh, okay. You actually didn't know. Sorry about that. Okay? Red blood cells. So they are isotonic with, meaning the inside of their membrane and the outside of their membrane have the same concentration of salts. And what is that number? I assume you guys don't know yet?
No.
It's 0.9%. Okay? So I want you to get the answer from the information I just gave you.
Creamy hypertonic solution.
Hypertonic. Okay, any other guesses?
It's an acid.
Isotonic? Isotonic. Okay. E. Lysis. Okay, so we have three options. Let's break this down, okay? First of all, 0.09% salt solution. Is that hypo or hypertonic or isotonic to what is inside the RBC? I said it's 0.9%, not 0.09. Okay, they might trick you with stuff like this, by the way. This is one of the only numbers you need to know in this course altogether, okay? So it is, the outside is hypotonic as compared to the inside. So now we know it's hypotonic, so it's not going to be B, C, or D. We're following? All good? So between A and E, what do we think? E. A, E. Okay. So I've come up with some goofy way to remember that. I hope it helps. So you know what happens when you cremate somebody? You know what happens when you cremate someone? Yes, you burn. You reduce it to ashes. So you're reducing its volume. You're shrinking down the volume. So when a cell shrinks, it crenates. It's not cremate. Please don't put cremate. I know you have MCQs, but just saying, okay? So crenation occurs associated with shrinkage of the cell, okay? And that is happening when the blood cells are placed in hypertonic solution. All good? Hypotonic solution, what happens? The blood cells are placed inside, that's what happened previously, okay? So the water moves from the solution into the blood cells.
So it's going to explode.
Yes. So it's going to grow, inflate, rip, burst. What is lysis? Breakage. Breakage of the cell? Lysis. So that is hypotonic. You guys, so if you get osmosis down, these are the only facts you need to know, the ones in bold. Crenation, hypertonic. Chemolysis, hypotonic. Isotonic is with 0.9% salt solution. I didn't wait by volume, but I'm not sure if you need to know that. Please work. Any doubts? Okay, we're almost there. Okay, 2 more slides. Alright, so what establishes osmotic pressure? Alright, so in a cell, alright, inside it you have proteins. And this is a fact, it's a universal fact that cells have anionic proteins. What does that mean? Negatively charged proteins, alright? So they cannot move out, or they cannot move across the cell because they're macromolecules, they're anionic proteins. Okay, so they stay inside, they have negative charge, okay? And on the outside you have other ions of positive charge. So it's gonna attract what? It's gonna attract opposite charge. Protons, positively charged ions into the cell, okay? It doesn't matter what nature or what element it is. It could be sodium, it could be potassium, it could be anything with a positive charge because the inside is negative, right? So in between though, in that process, something going on in the cell is preventing this movement of particular ions and allowing other particular ions. So what is that? We'll see in the next slide. And since that occurs, you have a high concentration of one type of ions in here, element, and the other ones are outside. So you're establishing a gradient, okay? So what is that process that's going on in the cell membrane that is preventing the movement of ions, particular ions? So this takes us back to solvation sphere, right? Sodium has the same charge as potassium?
No.
Also? Plus. It's not 2 plus, it's not 3 plus, plus. So same charge. But sodium, we should know that it's a smaller ion than potassium. So its charge density, you guys need to know this is important. The charge density is what affects how much of a solvation sphere an ion can attract. So sodium has a higher charge density, more charge per unit volume, and it's attracting more water molecules in a tighter and larger arrangement. Okay? So what happens in terms of potassium? It has lesser charge density, so it's doing what? Can someone tell me? Have I lost you guys? Sorry.
Can you repeat what?
Smaller solvation speed.
Smaller and looser. So sodium, smaller ion, same charge. So it has more charge per unit volume. So it attracts more water around it and more tightly. So the volume and strength of the solvation sphere is greater in terms of sodium. Potassium has a weaker and smaller solvation sphere. Why is this relevant, right?
That's charge density.
Right. But like, why am I talking about solvation spheres and the ions? Because for the ions that I was talking about, right, the ions outside of the cell to move in for the negative charge proteins, you're gonna need the solvation sphere to be removed. That's a cute little drawing I made here. Yeah. So you have a hydrated sodium solvation sphere, right? That is trying to enter the sodium transport protein in the membrane. So it's trying to cross the membrane, but it cannot because its hydration sphere is so tight and so large, right? So it needs to get rid of its solvation sphere to move in. On the other hand, potassium, are we following me? Pause. 1 second, okay? On the other hand, potassium has a looser and smaller sphere, so it can easily go in. So potassium ends up in a higher concentration inside the cell than sodium does. Sodium stays outside. Why? Because its solvation sphere is too big and too strong. Cool? So that establishes what gradient? Electrical gradient. Electrochemical, yes. So that is the gradient that is formed across the cell. So this is, why are gradients important? Does anyone know?
ETC, electron transport.
That's one example. So long story short, your body, your cells need to be in constant, dynamic. Like something needs to keep happening. We need to have ambition in life. Like you want to go somewhere, you want to do something, that keeps you moving. You're going to stay still, you're going to rot, whatever. If you have no ambition, no force, no gradient, no nothing, right? So what happens when your body, this is not in your textbook by the way, what I'm going to say. What happens when your body reaches a state of equilibrium? Like everything in your body is in equilibrium. You die, right? So you need this constant existence of a gradient, of something or the other that's not in balance. So that they keep moving to balance and just like gears, okay? If that makes sense. So that's what it is. Molecule ion transport, basically electrical gradients, anything to do with biochemical processes. Your life processes, and these are chemical. Last slide. I know I'm really funny. Sorry about that. Anyway, so what's this about? Osmotic pressure, right? So you have sodium staying on which side of the cell? It's in higher concentration inside or outside? Outside, right? So it's going to want to move in. Similarly, potassium is going to want to move out. And they do in very small amounts, right? Because they're that desperate. The gradient is so strong. They're going to want to cross. And they do. But the sodium-potassium pump uses ATP energy to push them back to where they belong. So, get out, right? Get out or get in, whatever. Right? That's what's happening. You keep the sodium-potassium pump keeps the sodium outside and the potassium inside and sodium outside. Cool. That's it. But one last question, or not. Have a great day. I want to make sure everyone gets it. If you don't, please don't hesitate to ask me. Okay.